Hello, my gentle and of course very modern apes. Welcome to another episode of Gutsick Gibbon. I'm Erica, or Gutsick Gibbon, and what we do here is we look at topics like paleoanthropology, so human evolution, biological anthropology, also human evolution, primatology, kind of human evolution, and sometimes we look at these things through the lens of debunking pseudoscience, but not today. Today we're going through a really cool new paper that was just released. So I guess I should clarify, the preprint is out on BioRx IV, but the preprint has also been covered by a Nature article, which I think all but assures its publication, so we're going to cover the preprint, keeping in mind that I guess it's some of the minor details are subject to change, but I read the preprint and the article that covers it, and they're the same. It's the same guy. So get excited, because today we're talking about the oldest genetic data ever recovered for a hominin thus far. And that hominin is Paranthropus. It's this guy. So Paranthropus is a super weird genus of hominin, meaning it's more closely related to modern humans than it is any other living great ape, and it's a sister group to genus Homo. So right now the idea is that you have Australopithecus, so like Lucy species, and two genera descend from that genus. You've got Paranthropus on one hand and Homo on the other. So genus Homo and genus Paranthropus both show up at like approximately 2.7, 2.8 million years ago, and while Homo is considered to be like an encephalizing generalist, as it were, that's a huge generalization, but you get the idea, Paranthropus is a little bit different. Paranthropus is a bipedal cow-like hominin, and when I say it's cow-like, I mean it's because this thing is eating grass, and it has the adaptations to boot for grass eating. So as you can see, it's got a really wide, weird-looking face with pushed forward or anterior zygomatics, those are the cheekbones. It's got a big sagittal crest right on the top of the head. Its molars are positively enormous, so this is a 3D print of a Paranthropus mandible, and here's a human side by side. So the molars of Paranthropus are like 4 to 5 times the size of those of a human, and it's got a massive amount of force that's able to be applied to these molars because of that huge sagittal crest, which is anchoring powerful temporalis muscles for grinding up its food, which is, weirdly enough, mostly grass. Although, again, if you'll remember, earlier this year, we got a report of a tool of tools and butchered animal bones being associated with Paranthropus remains. And granted, it was just a handful of molars, so we don't know for sure if Paranthropus was butchering or was being butchered. But that being said, it had the same capacities that Australopithecus would have had, and we don't really have an issue tentatively assigning tool use to Australopithecus. Anyways, so it's this weird sister group that seems to have adapted in an opposite direction to that of genus Homo, with big fat molars, molarized premolars, teeny tiny little nipping-like incisors, anterior zygomatics, and a sagittal crest. And we have genetic data for it now. Not only that, but this genetic data specifically has allowed us to deduce the sex of the specimens analyzed, as well as take a little peek into how these particular proteins, and thus these particular specimens, fit within the hominin family tree. The previous record for genetic material from a hominin hovered still around under 1 million years old. So the fact that we've got genetic material from these Paranthropus fossils, specifically they're coming from the enamel of the teeth, means we've more than doubled the age under ideal conditions that we can get genetic information from. And this information, to be clear, it's not DNA, and we're going to touch more on that in a second. Instead, what we've got is some sequenced proteins, but that can tell us a lot more than you might think. So here's the article in Nature summarizing the findings of this preprint titled Oldest Genetic Data from a Human Relative Found in 2-Million-Year-Old Teeth. Ancient protein sequences identify the sex of Paranthropus robustus fossils and hint at evolutionary relationships. So this is a specimen of Paranthropus robustus. There are 3 species of Paranthropus that are recognized, the oldest being Paranthropus aethiopicus, coming from East Africa, followed by Paranthropus robustus and Paranthropus boisei, who are from South Africa and East Africa again, respectively. So we're looking at South African specimens of Paranthropus, specifically teeth from Paranthropus robustus. So the article discusses previous records for genetic data, with the oldest preserved ancient DNA coming from Greenland permafrost at 2 million years of age, and for hominins specifically being Neanderthal stuff from, I think it's Cima de los Huesos at 400,000 years old. But proteins are a lot more robust, as they note here. It's resilient compared to DNA. And DeMarchi's team in 2016 was able to pull protein sequences from ostrich eggshells in Tanzania that were 3.8 million years old. Enrico Cappellini, who is our last author on the paper today, has also previously pulled proteins from 800,000-year-old remains belonging to Homo antecessor in Spain, as well as the Dmanisi material at 1.8 million years ago in Georgia. So they talk about the folks who are going to be involved and how they kind of got around to thinking to test the Paranthropus robustus teeth, which is found in Swartkrans, which is one of the classic sites in South Africa. They used a technique called mass spectrometry to analyze hundreds of amino acids in each sample's enamel, which is the outer layer of teeth. So just for the folks who might be wondering, the reason we don't test every single tooth or fossil that we can get our hands on for DNA and proteins is because it's a destructive technique. You have to destroy part of the fossil to actually analyze it for genetic material, which is why we tend to be very picky on what we kind of allow to go through that process. And it tends to be pretty difficult to get permissions, as I think is quite understandable and probably very appropriate. But the team is actually going to discuss in the paper itself some interesting alternatives in the future for perhaps looking at less destructive techniques to see if the genetic material is there in the first place before you actually go in and pull it, which is always going to be destructive. But at least this way you don't end up wasting specimens that you can't pull anything from in the first place. So they're going to talk about here how they managed to sex the specimens, because part of what is really interesting about this paper, we'll hop over here and look at it for a minute first before we appreciate what the summary says, is enamel proteins reveal biological sex and genetic variability within southern African Paranthropus. That's Paranthropus robustus. They were able to take these proteins and determine the biological sex of the specimens, which is super huge if you're, a person who studies sexual dimorphism like me, because it can help us tell how much variability within a species is due to differences between males and females, how much is perhaps the result of species variability in general. So the way that they actually sex the specimens is by looking at a specific protein called amelogenin Y, which is produced by a gene on the Y chromosome. Its presence in two of the samples allowed the researchers to conclude that the teeth belonged to males. One of these had been previously attributed to a female on the basis of its small size. So this is a problem inherent to an order of mammals being primates that can be sexually dimorphic, is sometimes we look at two specimens, one is big and one is small, and we say, these are two species, and sometimes we look at them and we say it's a male and a female of a single species, and it's really hard to tell which is appropriate sometimes. The other two teeth lacked signs of amelogenin Y and contained the X chromosome version of the protein, leading the authors to deduce that the specimens were probably from females. It's a good thing that they're looking for the presence of the X chromosome version, because some modern human populations, and indeed Neanderthal specimens, like Neanderthal individuals, have actually undergone mutations where they've lost the amelogenin Y protein despite being male otherwise. So it's good that we're not using this as like the sole criteria for sexing the specimens. So they talk about how many amino acids they sequenced, around 400 of the same amino acids in all 4 samples. This allowed researchers to build a simple evolutionary tree confirming that Homo sapiens, Neanderthals, and hominins found in Siberia called Denisovans that lived in the past 200,000 years are all more closely related to one another than they are to the 2 million year old Paranthropus. Okay, yeah. I should hope so. Any other relationship would have been a big surprise, says Duca. Yeah, I would imagine. In one enamel protein, researchers found sequence differences between the Paranthropus remains, potentially reflecting variability within the species. Building an evolutionary tree from genetic data of such old remains can be considered a potentially transformative breakthrough for paleoanthropology, Cappellini and his colleagues write in the preprint. They add that ancient protein studies could improve the understanding of where creatures such as Australopithecus afarensis, of which there are many fossil fragments, and the more complete specimen known as Lucy sit in the hominid family tree. Other scientists say the jury is still out on whether ancient proteins will help bring consensus to the picture of hominid evolution, which is currently built largely from the shape of bone, so from morphology. They talk about how there's a long way to go with proteomics in general, but that as ancient proteomics expands, researchers will be able to balance the benefits against the costs of destructive sampling and the lab in question is actually working on non-destructive methods to screen fossils for their protein content, sparing samples that wouldn't have yielded good data. It's important that the risk of failure is considered when taking decisions about sampling the fossil record, he says. So this is kind of what we were talking about earlier. So now that we've read sort of the summary here and you guys have a good background, let's look at what was actually done here in this absolutely phenomenal preprint. As always, we start with the abstract. The evolutionary relationships among extinct African hominin taxa are highly debated and largely unresolved, due in part to a lack of molecular data. True. Even within taxa, it is not always clear, based on morphology alone, whether ranges of variation are due to sexual dimorphism versus potentially undescribed taxonomic diversity. For Paranthropus robustus, a Pleistocene hominin found only in South Africa, both phylogenetic relationships to other taxa and the nature of interspecific variation are still disputed. This is all true, right? We don't know how much diversity is due to sexual dimorphism or not, and where Paranthropus robustus sits in relation to the rest of the hominins could use a little bit of rounding out with molecular data, to say the least. Here we report a mass spectrometric, here we report the mass spectrometric sequencing of enamel proteomes, from 4 2-million-year-old dental specimens attributed morphologically to Paranthropus robustus from the site of Swartkrans. Now, my understanding later on in the paper is that they pulled for premolars and molars, which, as I hope was demonstrated earlier, are pretty easy to clock as a member of genus Paranthropus, so there's little ambiguity there. The identification of MLE-specific peptides in semi-quantitative MS data analysis enabled us to determine the biological sex of all specimens. Our combined molecular and morphometric data also provide compelling evidence of a significant degree of variation within South African Paranthropus, or Paranthropus robustus, as previously suggested based off of morphology alone. Finally, the molecular data also confirm the taxonomic placement of Paranthropus within the hominin clade. This study demonstrates the feasibility of recovering informative early Pleistocene hominin enamel proteins from Africa. Crucially, it also shows how the analysis of these proteins could contribute to understanding whether hominin morphological variation is due to sexual dimorphism or to taxonomic differences. We anticipate this approach will be widely applied to geologically comparable sites within South Africa and possibly more broadly across the continent. So they add a little bit of background on Paranthropus and discuss how this might help resolve some phylogenetic relationships. So specifically here, they say even within Paranthropus, the phylogenetic relationships among the 3 currently identified species has been a subject of considerable discussion. Most researchers consider Paranthropus taxa to be monophyletic. However, morphological similarities between Paranthropus robustus and Australopithecus africanus in a South African context, as well as between Paranthropus aethiopicus and Australopithecus afarensis within an Eastern African context, have raised the possibility of paraphyly or even admixture between species. So there are 2 kind of scenarios bopping around for Paranthropus. In one scenario, you have Australopithecus, the genus, evolve in East Africa, take the Green Highway down to South Africa, and evolve into Australopithecus africanus and Australopithecus sediba, etc. And then the same thing happens to Paranthropus. It emerges in East Africa, a species takes the Green Highway south to South Africa, where it evolves into Paranthropus robustus. There is another alternative idea, though, which is that in that first situation, the Australopiths that make their way down to South Africa and evolve into Australopithecus africanus then evolve into Paranthropus robustus in South Africa, while the Australopiths that stay in East Africa convergently yield Paranthropus aethiopicus and Paranthropus boisei there. That would make Paranthropus a paraphyletic group. This is something that work with proteins could potentially elucidate, although probably not in this paper since we're exclusively looking at things that are designated as Paranthropus robustus. We're not looking at Australopithecus africanus or any of the East African members of Paranthropus. The authors also mention a third idea, which involves admixture. It's a huge mess with hominins, because hominins are not picky, as we know, being hominins. So I should have just done the previous section here when we've got this nice diagram, but as you can see, we've got a really cool figure. C shows us the 4 specimens in question, which are all molars or premolars, and we have this nice diagram of Africa that shows us where Paranthropus can be found. So in the situation where Paranthropus is not paraphyletic, we see Australopithecus evolve here in East Africa. Some members of Australopithecus take the Green Highway, which is this area here on Southeast Africa that periodically is green and periodically is difficult to navigate. When it's green, hominins can make it south, and when it's not, they can't. So when it's green, some Australopiths make their way south, where they evolve here in South Africa into things like Australopithecus africanus and Australopithecus sediba. Then Paranthropus does the same thing, evolves here. Some members take the green highway south, where they evolve into Robustus down here and into Aethiopicus and Boisei up here. In Paranthropus's paraphyletic, we see Australopithecus evolve here in East Africa, same situation, but the members that make it down south and become Australopithecus africanus, or at least yield it down in South Africa, also yield Paranthropus robustus. And then the same thing happens in East Africa, where the Australopiths here yield Paranthropus boisei and Paranthropus aethiopicus. That would make Paranthropus paraphyletic, which is something of a no-no. So they talk about here the importance of genetic data, how it provides insights into how hominin groups relate to one another. They talk about where they got the 4 teeth for hominin fossils, originating from Swartkrans, which is approximately 40 kilometers northwest of Johannesburg in South Africa, what they're dated to between 1.8 and 2.2 million years ago. They talk about how they maximized their amino acid coverage and how they made sure that, in fact, these were legitimately from Paranthropus, how they ensured that there was not contamination and things like that. And then we get down here to specimens SK850 and SK835 were unambiguously identified as male Paranthropus individuals based off of the observation of multiple overlapping amelie-specific peptides. Then they talk about how the absence of the amelie-specific peptide can't definitively lead to a female identification, and they're going to talk about that a little bit more in the description as well. Moving down to the phylogenetic reconstruction, which is also something I'm interested in. Can we definitively show that Paranthropus is a hominin and not like an outgroup to the hominins, or potentially even more closely related to like gorillas or chimps? This is just good to check off of our list here. So they say to attempt a molecular-based phylogenetic reconstruction, the assembled protein sequences from all 4 Paranthropus specimens were aligned to their orthologs from large reference data sets, including multiple individuals of extant and extinct hominids. Based off of the phylogenetically informative sites identified, all 4 Paranthropus sequences were closer to those in the Homo clade than to any other primate. So definitively a hominin. Genetics tells us that they're a hominin. We can check that off of the list. And you know, genetics is, of course, the most important thing to consider when we're looking at relationships between hominins. In the 4 Paranthropus individuals, we detected a total of 17 hominid species informative single amino acid polymorphism, or SAPs. So there are 17 specific amino acids in their sort of string of amino acids. Out of the total number of amino acids, there are 17 that are informative to the placement within hominids. The others just do not distinguish within hominids specifically. They are maintained by all of the members of Hominidae. Only 2 of which showing an allelic state different from present-day humans, Neanderthals, and Denisovans. So of the hundreds of amino acids that these researchers were specifically looking at, only 17 could be species-specific. So only 17 amino acids of all of those strings were different within various species of hominids. The rest all hominids share. And of those 17 that could distinguish on the basis of species, like on the species level, only 2 amino acids differed between the Paranthropus specimens looked at and the group of humans, Neanderthals, and Denisovans. That's how much we differ in this string of amino acids. They are definitively hominins, ladies and gentlemen. So of these 2 that separate Paranthropus from the group of later genus Homo, from the members of genus Homo looked at, one of them discriminated genus Homo, the members of genus Homo, from Paranthropus, with Paranthropus sharing the ancestral state with non-hominins. So Paranthropus has the version that modern-day great apes have, whereas genus Homo, the members of genus Homo looked at, have the derived condition. And the other one was different, even within Paranthropus. So it separated the 4 Paranthropus specimens from the other primates looked at, including genus Homo, but it was also slightly different within Paranthropus. So what they say at the bottom here is, accordingly, our results indicate that this substitution most likely occurred independently in Paranthropus, but did not fixate within all wider populations or groups of this genus, which is interesting in and of itself. It suggests that there's some species variation going on here. So within species variation is similar to that observed on ENAM 137 is expected for any large enough data set of amino acid sequences, but may be unexpected for a sample size of 4 individuals. So they look at this a little bit deeper, And they basically noticed that you could potentially get this type of variability in 4 sampled human individuals, but Paranthropus' population dynamics might have been a little bit different anyway. So they're saying any taxonomic conclusions for within Paranthropus, not between Paranthropus and other hominins, but within Paranthropus, on the basis of this genetic variation alone seems premature. So they're saying that you can't look at the variation of this one specific amino acid within the Paranthropus sample and say that this justifies like new species or anything like that. They say to explore the taxonomic placement of the Paranthropus individuals, we utilized our aligned reference data sets to generate phylogenetic trees via maximum likelihood in a Bayesian approach. The phylogenetic reconstructions place Paranthropus individuals as outgroups to the clade containing present-day humans and available Pleistocene hominins from Eurasia. So this is our members of genus Homo. All of these individuals, including Paranthropus, form a clade to the exclusion of the other members of present-day Hominidae. 3 of the Paranthropus specimens form a clade, while the third appears to be more distantly related to them. These results were also independently validated by repeating the phylogenetic analysis using an alternative workflow and reference dataset. So the discussions and the broader implications just kind of sum things up. So initially they discussed like why is it we might be able to pull proteins from this enamel and where might it not be as applicable, where might proteomics be less applicable. They say the specimens that we studied were recovered from cave sediments mostly composed of remobilized soil from outside the cave. Altogether, their rapid accumulation through episodic flash floods and the relative aridity coupled with extensive cementation favorably contributed to the preservation of the proteins within fossil teeth. Whether or not protein preservation would be comparable in other early hominin-bearing deposits, including open-air sites such as those found in other places in Africa, is an open question, and future work should focus on testing the feasibility of paleoproteomic work or paleoproteomic analysis while minimizing damage to precious African heritage. They talk about the nature of how they actually pulled the proteins from the teeth themselves, and they talk about how we are looking at a specific set of genes here. We're not looking at a full genome or anything like that. So any individual gene trees can only be inferred partially, as we can only observe mutations that have resulted in amino acid substitutions located in peptides covered by mass spectrometry, and basically saying that the tree should be considered tentative, at least at this point. But despite these caveats, the recovery of the 2-million-year-old phylogenetically informative genetic material in African hominids can be considered potentially transformative with regard to paleoanthropology, and I would say that that's absolutely true. They talk a little bit about the specific genes, again, and the variability within species and the ability to sex the samples, saying that they're confident identification of both male and female Paranthropus individuals. And then finally, they finish just by touching on how huge this is if it can be applied more broadly. The coherent results obtained from this study, combining molecular and morphologic data, have profound implications for addressing such long-standing controversies surrounding the nature and extent of Pleistocene hominin diversity. I just absolutely cannot wait to see the broader application of the methodology within this study. I hope we pull proteins from just about every hominin out there, and I hope we figure out a less destructive way to do it as well. As a side note, given I dunk on like young earth creationists and pseudoscientists in general on this channel, this is really bad news for them, right? Genetic work that shows that Paranthropus nests within the hominins is going to be a hard one to reckon with for a great many of these guys that are sort of subscribing to this idea of true human exceptionalism, that there's something about humans morphologically, genetically, biologically as a whole that separates us from the other apes, both in space and in time. This shows us that, yeah, Paranthropus is more closely related to us than it is to chimps or gorillas or any other living great ape. And I think that one's going to be a real doozy of a pill for them to swallow. Thank you so much for watching, you guys. This was a really exciting one. If you like what I do, you can support me in a free way by liking, commenting, and subscribing, or alternatively, you can head on over to my Patreon and become a patron. Patrons get their names at the end of videos, and they sometimes get early access to videos. And so, my gentle and of course very modern apes, I'll see you real soon in the next one.